We're talking from the smallest coil of DNA inside the nucleus all the way out to the largest, most complex metabolic cycles that generate every single bit of energy we use.
It's truly a comprehensive set of, you could almost call them engineering schematics.
Right.
When people hear biochemistry, they often brace themselves for just endless lists of confusing names and pathways.
Right. Memorization.
Exactly. But what we have here is different. It's all organized around systems, and I think most critically, around the diagrams that connect all of those systems.
Absolutely. And that's our mission today. We want to move past just the memorization and give you, the learner, a really structured, deep understanding of these foundational molecular machines. We're trying to bridge the gap between genetics, cellular structure, nutrition, and metabolism.
And the scale is just staggering, as you said. We're starting at the microscopic level, how DNA is physically organized, and then we're going to soar out to trace how fatty acids get shipped across the entire body.
And even dive into how a simple, single vitamin deficiency can completely shut down an entire energy production pathway.
I like to think of it this way. We're moving from looking at a house to looking at the architectural blueprints. And not just the blueprints, but the plumbing diagrams, the electrical wiring schematics too.
Okay, I like that analogy.
Because the blueprints don't just tell you what parts are there. They show you how they interact. And crucially, where one small defect, a faulty enzyme, a missing cofactor, a misplaced protein, can lead to total system failure.
Which is what we see as disease.
Precisely. This depth is what turns what feels like an overwhelming collection of facts into a single interconnected functional map of the body.
So let's start at the absolute foundation then, inside the nucleus. This is the ultimate storage problem.
It really is.
So we're in part one, the blueprint. And first up is packaging the genome, chromatin structure and control.
Yeah, so DNA is incredibly long. If you stretched it out, it would be meters long.
Yeah.
And it has to fit into a space smaller than a speck of dust.
How does that even work?
The sources illustrate this concept beautifully by focusing on something really simple. Electrical charge. We all know DNA is negatively charged.
Right, because of all the phosphate groups on its backbone, they're all negative.
Exactly, and that negative charge is the key. It's the signal for interaction. So to manage this incredible length, the DNA wraps itself around these positively charged proteins called histones.
Okay, so a positive spool for a negative thread. But why are the histones positive? What makes them that way?
It's their amino acid composition. Histones are incredibly rich in two specific basic amino acids, lysine and arginine.
Ah.
And the side chains on lysine and arginine at normal body pH carry a net positive charge. So they are the perfect molecular spool for that negatively charged DNA thread. It's a fundamental electrostatic attraction.
Right. And the diagram shows that when the DNA wraps around, twice? Around a core of 8 of these histones.
An octamer, yes. 2 copies each of 4 different histones, H2A, H2B, H3, and H4.
When it does that, you form the basic unit, the nucleosome. So that's level 1 of compaction.
But the structure doesn't stop there. That would still be too messy. So those individual nucleosomes, which look like beads on a string, are then linked together by another specialized histone called H1.
Okay, so H1 is like the staple.
It is. It acts just like a staple. It clamps the DNA onto the histone core and helps coil all those nucleosomes together, stabilizing the entire chromatin fiber. And this entire structure isn't just for packing. This is the body's primary switch for controlling which genes get expressed.
And this control, this is where we get the 2 fundamental states that are always discussed. Heterochromatin versus euchromatin.
Precisely. Let's start with heterochromatin. It's the highly condensed state. If you look at it under an electron microscope, it appears super compact and dark. It's transcriptionally inactive.
It's basically in storage mode. Locked down, silent.
You got it. The cell cannot read the genes in those sections efficiently. They're packed away for later, or maybe forever, in that cell's life.
But then you have euchromatin, and the key is right there in the name. Eu means true.
So it's truly transcribed. This is the active form. It's much less condensed. It looks lighter on an EM, and these are the genes that are currently being read and expressed by the cell. This distinction is really the engine of cellular identity. A liver cell and a brain cell have the same DNA, but what's in euchromatin versus heterochromatin is completely different.
So how does the cell actually flip that switch? The source highlights the critical role of epigenetic modifications. These are changes that don't alter the DNA sequence itself, but they drastically alter how accessible it is.
The 2 core methods detailed here are DNA methylation and histone modification. Let's start with DNA methylation. This process usually happens at specific regions that are rich in cytosine and guanine bases, We call them CPG islands.
And they're often located right near the promoters of genes, the on switch.
Exactly. So when DNA gets methylated in those areas, it fundamentally silences gene transcription.
It's essentially putting a permanent padlock on that gene promoter. It physically blocks the necessary transcription factors from being able to bind.
Right. And we see the tragic consequences of this process going wrong in disorders like Fragile X syndrome. That involves an abnormal expansion of CGG repeats in a specific gene, which leads to hypermethylation. And that silences the FMR1 gene, causing the syndrome.
Wow. So contrast that silencing mechanism with the other side of the coin, histone acetylation.
Histone acetylation is for activation. It's the on switch. Special enzymes come in and add an acetyl group to the histone tails, and this neutralizes their positive charge.
And if you neutralize the positive charge on the histone,
The strong attraction between the histones and that negatively charged DNA just weakens. It lets go.
so the coil loosens.
The DNA uncoils, it loosens up. And this opening of the chromatin allows all the transcriptional machinery to get in there and access the DNA, leading to a massive increase in transcription.
It's fascinating that the source points out a direct systemic link here. Thyroid hormone synthesis actually works by activating histone acetylation.
It does. It ensures that critical metabolic genes are expressed to maintain the body's hormonal balance. It's this beautiful, clean line from a systemic signal, a hormone, right down to the mechanical coiling of DNA.
And the opposite, of course, is histone deacetylation. Taking those acetyl groups off tightens the coil back up, shutting down transcription. The source links the specific decrease in gene readability to the altered gene expression seen in Huntington disease. So this isn't about mutations, not always. It's about changing the mechanical state of our genetic material to regulate life itself.
Exactly right.
So from the packaging, let's move to the actual building blocks, the nucleotides.
Okay, so a nucleotide is the full assembly. You've got the nucleoside, which is the nitrogenous base, plus the sugar, either deoxyribose for DNA or ribose for RNA.
And then you add the phosphate group.
And you add the phosphate group. And these components are all linked together in a growing DNA or RNA strand by a crucial 3' to 5' phosphodiesterase bond. That establishes the all-important 5' to 3' directionality of everything that follows.
And we break the bases themselves into two structural categories. You have the purines adenine A and guanine G. They're the larger ones, composed of 2 rings.
And we remember them with the mnemonic pure as gold.
Pure as gold. And the others?
The pyrimidines, cytosine C, uracil U, and thymine T. They're the smaller ones, just a single ring. The mnemonic for that is CUT, the pyramid.
CUT, the pyramid. Got it. And this structural difference, the size, it matters immensely when it comes to the stability of the DNA helix itself.
It's critical. The source highlights a really important detail. GC pairs are connected by 3 hydrogen bonds.
Whereas AT or AU pairs only have 2.
And that third hydrogen bond in the GC pairing makes a huge, huge difference. It means DNA regions that are rich in GC content are thermodynamically much more stable. They have a significantly higher melting temperature.
So if a segment of DNA needs to be kept really tightly bound, like at the start of a very important gene, it's probably going to be GC rich.
Very likely, yes.
Okay, so let's look at the construction. We need specific amino acid precursors to build these rings. For purine synthesis, it's glycine, aspartate, and glutamine, the JAG amino acids.
Right. And for pyrimidine synthesis, you need aspartate and carbamoyl phosphate. And you should put a pin in carbamoyl phosphate because we are definitely going to revisit that when we discuss the urea cycle later on.
Good to know. So these amino acids are basically the raw materials that the cell stitches together to make the rings.
They are. But when we talk about building blocks, we also have to talk about recycling. The purine salvage pathway is the body's highly efficient, preferred method for reusing existing purine bases instead of making them from scratch every time.
And when this recycling pathway fails, the whole system just clogs up dramatically.
And the prime example of that failure is Lesch-Nyhan syndrome. It's a devastating X-linked recessive condition. The defect is in one specific enzyme, HGPRT. Hypoxanthine guanine phosphorybosyltransferase.
That's a mouthful.
It is. But the diagram in the source showing the salvage pathway makes the failure immediately clear. If HGPRT is defective, it can't salvage hypoxanthine and guanine.
So instead of being recycled,
They get shunted towards degradation. And the end product of purine degradation is uric acid. So you get a massive overproduction and accumulation of uric acid, which results in hyperuricemia.
And the clinical consequences are they're catastrophic. It's not just the biochemical buildup, it's profound neurological dysfunction.
Exactly. Patients exhibit severe intellectual disability, spasticity, aggression, and the notorious self-mutilation, sometimes biting off their own fingers or lips. It's terrible. And because the core defect is an overproduction of uric acid, the main goal of treatment is to lower that uric acid level, usually using inhibitors like allopurinol or febuxostat.
It's just incredible that one enzyme failure causes such a complete systemic meltdown. And speaking of fatal recycling failures, we also have to talk about adenosine deaminase, or ADA, deficiency.
Yes, ADA is required for the degradation of adenosine. A deficiency here is lethal to the immune system
Why the immune system specifically?
because the lack of ADA leads to the accumulation of a toxic intermediate, deoxyadenosine triphosphate, or DATP. And for reasons we don't fully understand, DATP is specifically toxic to rapidly dividing B and T lymphocytes. It just kills them off.
Which causes one of the most severe forms of inherited immunodeficiency, autosomal recessive severe combined immunodeficiency, or SCAD, the bubble boy disease.
That's the one. So these two disorders, Lesch-Nyhan and ADA deficiency, are just powerful examples of how a failure in a seemingly mundane recycling pathway can have direct, lethal consequences on the nervous system and the immune system.
Okay, the blueprint is organized, the blocks are ready. Now, how is the blueprint actually copied? The sources provide a critical diagram of the replication fork, illustrating this semi-conservative process that always proceeds in the 5' to 3' direction.
And this truly is a complex ballet of molecular machinery. It's not just one enzyme, it's a whole crew. And defects in any of the key players lead to specific genetic instability syndromes. Let's start with the unwinding action.
That's the job of helicase, right? It unwinds the DNA template, separating the two strands. It's the unzipper.
It's the unzipper. And if this enzyme is defective, as you see in Bloom syndrome, the DNA can't replicate stably. This leads to massive chromosomal instability and a much higher risk of cancer.
As soon as the strands are separated, I see these single-stranded binding proteins rush in.
Yeah, they prevent the strands from snapping back together or from being degraded by other enzymes. They're like little doorstops. But the unwinding creates this massive stress. The DNA ahead of the fork starts overwinding, like if you twist a rubber band too much.
Ah, right, the supercoiling problem.
Exactly. And this is where DNA topoisomerases come in. They are absolute genius enzymes.
So they're the tension release valves.
That's a perfect analogy.
Yeah.
They relieve the twisting stress, the supercoiling, and the source differentiates between topoisomerase the first, which makes a controlled, single-stranded break to let it unwind, and topoisomerase the second, which makes a full, double-stranded break.
Why is that distinction so important?
Because we can exploit it clinically. A lot of our most essential anti-cancer drugs target these specific enzymes. Irinotecan and topotecan inhibit topo first. Etoposide and tenofoside inhibit topo second.
So the drugs let the cancer cells start to replicate, but then they prevent the topoisomerases from relieving the tension, and the DNA just shatters.
It shatters under the strain. You're leveraging the fact that rapidly dividing cancer cells absolutely need these enzymes to survive. It induces lethal, irreparable stress on the cancer cell's genome.
Okay, so to actually start synthesizing the new DNA, we need Primus to lay down a short RNA primer. DNA polymerase III can't start from scratch.
Correct. Then DNA polymerase III does the heavy lifting. It elongates the leading strand continuously and creates those short Okazaki fragments on the lagging strand, all in that 5' to 3' direction.
And we can't forget the cleanup crew. In prokaryotes, DNA polymerase II performs a vital function.
It does. It grades that RNA primer and fills in the resulting gap. It uses its unique 5' to 3' exonuclease function to do that. It's like a little snowplow that removes the RNA and lays down DNA behind it.
And finally, DNA ligase comes in and seals the deal. It joins all those Okazaki fragments together by catalyzing the formation of the final phosphodiester bonds.
And one last piece of the puzzle, telomerase. As the chromosome shortens with every round of replication, telomerase steps in. This special enzyme adds repetitive TTT sequences to the ends of the chromosomes, the telomeres.
This is highly active in stem cells and progenitor cells, but critically, it's upregulated in most cancers. It's part of what gives them their endless, immortalized growth potential.
Absolutely. But the cell is constantly battling errors. The diagram illustrating DNA repair mechanisms is just as important as the one for replication. It shows that the body doesn't just need speed, it needs precision, and it has multiple redundant systems to achieve it.
So let's compare the primary mechanisms. Let's start with nucleotide excision repair, or NER. What kind of damage is it specialized in fixing?
NER is the system for removing bulky lesions, specifically things like the pyrimidine dimers that are caused by UV light exposure from the sun.
Okay, so sunburn damage.
Exactly. It recognizes the distortion that these bulky adducts create in the helix. It doesn't just fix the one bad base. It excises a whole large segment around the damage and then synthesizes new DNA to fill the gap.
And the clinical link here is huge. A defect in NER causes xeroderma pigmentosum.
Yes, characterized by severe photosensitivity and a dramatically increased lifetime risk of skin cancer. The sources also specify that NER occurs predominantly in the G1 phase of the cell cycle before replication begins.
Okay, then you have base excision repair, BER. This sounds simpler.
It is simpler and quicker. It removes a single damaged base. It's like a repairman addressing one small leak instead of replacing a whole section of pipe. It's crucial for fixing damage from spontaneous or toxic deamination events, which happen all the time.
And finally, mismatch repair, or MMR. If NER handles the bulk damage and BER handles the small chemical damage, what is MMR's role?
MMR is the post-synthesis quality control system. It's the proofreader. After the DNA is copied, this system scans the newly synthesized strand for misspaired nucleotides that DNA polymerase missed.
And how does it know which strand is the right one?
That's the genius of it. It can tell the difference between the old template strand and the new error-prone strand. So if it finds a mismatch, it preferentially repairs the new strand, knowing the old strand is the correct template.
And a defect in MMR is the cause of Lynch syndrome.
Yes, formerly known as hereditary non-polyposis colorectal cancer, or HNPCC. This is why patients with Lynch syndrome have such a high cancer risk. Their basic quality control system for DNA copying is broken. And this process happens predominantly in the S phase, right after replication finishes.
And for the most severe event, a double-stranded break, the body has non-homologous end joining, which is a quick, dirty repair that just glues the fragments together, often losing a few nucleotides.
Right, it's an emergency fix. For high-fidelity repair, it uses a much more complex process called homologous recombination, which uses a complementary strand as a perfect template. The whole system is just layered and complex, ensuring that the genetic blueprint, despite constant threat, remains stable.
Okay, we've established the blueprint. Now let's leave the nucleus and step onto the factory floor of the cytoplasm, where the proteins are actually made, modified, and shipped out.
Part 2, the factory floor.
And we start with protein trafficking in organelles, referencing the cell trafficking diagram.
The cell is an astonishingly organized factory. We have to follow the product, the protein, from its synthesis site all the way to its final destination.
So the journey begins at the rough endoplasmic reticulum, the RER.
Right. This is the synthesis site for all proteins that are destined to be secreted out of the cell, incorporated into the cell membrane, or delivered to other organelles like the lysosome. This is also where the initial sugar chains are added, a process called N-linked glycosylation.
But not all proteins are made there, right?
No. Proteins that are destined to just stay in the cytoplasm, or those heading for the peroxisome and mitochondria, are synthesized on free ribosomes just floating in the cytoplasm.
Okay, so once the RER synthesized proteins are finished, they head to the Golgi apparatus.
Yes, the Golgi. This is the cell's sophisticated post-translational modification and sorting center. I always call it the cellular post office.
The post office, okay. And the source emphasizes a critical sorting mechanism here. For proteins that are destined for the lysosome, the Golgi has to add a very specific tag.
It does. It has to add mannose-6-phosphate, or M6P. This M6P tag is the unique lysosomal zip code that ensures proper delivery.
So what happens if the post office fails to put that zip code on the package?
Then you get a severe disease called I-cell disease or inclusion cell disease.
Wait, so I-cell disease is caused not by a bad enzyme inside the lysosome, but by the post office failing to put the stamp on the package. That's a perfect way to think about it.
That is exactly what it is. It's an inherited lysosomal storage disorder where the Golgi fails to phosphorylate those mannose residues into the M6P address tag.
So the lysosomal enzymes, they just get lost in the mail?
They get secreted extracellularly, they're shipped out of the cell by mistake. And inside the cell, the lysosomes never get their digestive enzymes. So cellular debris accumulates, forming these large inclusion bodies. It causes severe developmental delays and structural issues, and it's tragically often fatal in early childhood.
Wow. And to ensure accurate delivery throughout the cell, the source details 3 main vesicular transport systems. These are like the cell's internal courier service, all visible in the trafficking diagram.
Right. You have COPI, which handles retrograde transport shipping things backward. It's usually retrieving escaped RER proteins from the Golgi and sending them back to the RER where they belong.
One-way trip. And CoPI.
CoPI handles anterograde transport moving forward, from the RER to the cis-Golgi network.
And the third one is clathrin.
Yes, clathrin. It forms this recognizable basket-like coat around vesicles that are used for endocytosis. For example, when the cell needs to internalize hormones, or, most famously, when it takes up LDL cholesterol using the LDL receptor.
Okay, before we leave the organelles, we need to talk about peroxisomes. They seem like the cell's waste management and cleanup crew.
That's a great description. They are crucial detoxification and catabolism centers. Their specific jobs include breaking down very long-chain fatty acids, VLCFA, the alpha oxidation of branched-chain fatty acids, and the crucial first step of ethanol detoxification.
So if the cleanup crew is defective, the waste just builds up. And that's Zellweger syndrome.
Yes, it's an autosomal recessive disorder where the peroxisomes are either defective or they just fail to form entirely. The accumulation of those very long-chain fatty acids in the brain leads to profound neurological issues, hypotonia, jaundice, seizures, and often death within the first year. It's a powerful illustration of how the simple inability to break down oversized lipids can just cripple neurological development.
Okay, moving outside the cell. The extracellular matrix provides all the structural integrity, largely via collagen and elastin. And collagen is the most abundant protein in the body, providing incredible tensile strength.
The sources categorize 4 major types by their location, which really emphasizes their structural specialization. Type 1 is the strongest and most widespread. It's in bone, tendon, dentin, and fascia.
Type 2 is cartilage.
Type 3 forms reticulin, which you find in blood vessels, the uterus, and granulation tissue, the stuff of healing. And type 4 forms the fine mesh of the basement membrane.
The synthesis of collagen is a spectacular illustration of the necessity of cofactors. The source diagram breaks this multi-step process down into a really intricate assembly line.
The initial step is synthesis, inside the RER. It's a translation of the alpha chains, which are characterized by that repetitive glycine XY amino acid sequence. But the next step is the crucial one, post-translational modification, hydroxylation of proline and lysine residues.
And this hydroxylation is absolutely essential for the chains to coil properly and form that stable triple helix. And here's the nutrition link. This step requires vitamin C.
It absolutely requires vitamin C. Without it, the collagen is unstable and easily degraded. This leads directly to scurvy,
Which is characterized by easy bruising, bleeding gums, poor wound healing, all signs of compromised connective tissue strength throughout the body.
Exactly. So following hydroxylation, there is glycosylation and the formation of the triple helix structure, which we call procollagen. This soluble procollagen is then exported out of the cell via exocytosis.
Once it's outside, it undergoes proteolytic processing. Specialized enzymes cleave off the soluble disulfide-rich terminal ends. This is what turns the soluble procollagen into insoluble tropocollagen, which is ready for assembly.
And the final step, cross-linking, reinforces the whole structure. This is what locks the tropocollagen fibrils together, giving collagen its immense, load-bearing tensile strength. But this step requires the enzyme lysyl oxidase.
And lysyl oxidase requires copper as a cofactor.
It does. So just as vitamin C deficiency leads to scurvy, copper deficiency causes Menkes disease.
Right. The X-linked recessive disorder with impaired copper absorption.
Yes, and that leads to defective lysyl oxidase activity. So the lack of proper cross-linking manifests dramatically.
Yeah.
You see brittle, sparse, kinky hair, and dangerously fragile blood vessels, which can lead to cerebral aneurysms.
That's a detailed look at how structure requires continuous maintenance. But structural defects can also be purely genetic. Osteogenesis imperfecta, or brittle bone disease, is a genetic defect in type I collagen itself.
Right. And the findings tell a story of systemic connective tissue failure. You see multiple fractures from minimal trauma, blue sclerae, where the underlying choroidal veins show through the thin connective tissue of the idental imperfections, and progressive hearing loss.
Then you have Ehlers-Danlos syndrome, EDS, which involves faulty collagen or collagen processing, commonly type 3 procollagen.
And these patients have that characteristic hyperflexibility of their joints, hyperelastic skin that is easily damaged, and a tendency toward easy bruising. The vascular type, which is linked to type 3 collagen, is the most serious because of the risk of organ and arterial rupture.
And we also have to address elastin, which provides stretch and recoil, crucial for the lungs and large arteries. The source connects this immediately to Marfan syndrome.
Right, where the defect is in the FBN1 gene, which codes for fibrillin 1. Fibrillin 1 acts as a crucial scaffolding upon which elastin is deposited. Without good scaffolding, you get weak elastic tissue.
And the key symptoms of Marfan syndrome are structural issues in tissues rich in fibrillin, specifically aortic root dilation and long limbs. But we have to note the specific lens dislocation.
Yes, this is a key diagnostic point. In Marfan, it's an upward and temporal displacement. The source demands we contrast this with the defect in homocystinuria, an autosomal recessive condition.
Homocystinuria often mimics Marfan's tall stature, but it's caused by a defect in cystathionine synthase, leading to the accumulation of homocysteine.
And its lens dislocation is diagnostic. It causes the lens to be displaced downward and nasally. That one single clinical detail can differentiate the two conditions and highlights the precision required in diagnosis.
Okay, before we transition into the energy economy, let's briefly cover the sophisticated tools used to study these intricate cellular processes. The sources dedicate significant time to high-yield lab techniques that rely entirely on the biochemical principles we've been discussing.
Let's begin with the engine of molecular detection, polymerase chain reaction, or PCR. The diagram shows how this process rapidly amplifies DNA segments by cycling through three distinct temperature steps.
First, denaturation. You heat the double-stranded DNA to about 95 degrees Celsius to separate the strands.
Then, annealing. You cool the mixture down to allow the specific DNA primers to bind to their complementary sequences on the template strands.
And third, elongation. You raise the temperature slightly so a heat-stable DNA polymerase, like tachypolymerase, can come in and synthesize the new DNA strand, starting from the primer.
The power of PCR is its exponential nature. Each cycle theoretically doubles the amount of DNA, allowing scientists to detect even trace amounts of specific genetic sequences or pathogens. It's an indispensable diagnostic tool.
Then there is the revolutionary technology of genome editing, CRISPR-Cas9. It feels like science fiction, but it's really just a bacterial defense system that we've repurposed to edit genes with incredible precision.
Cas9 is a sophisticated endonuclease. It's a molecular scissor. And its power comes from the guide RNA, or gRNA, which acts like a GPS system. It targets and leads the Cas9 enzyme to a specific complementary DNA sequence. Once it finds the target, Cas9 cuts the DNA double helix.
And the outcome then depends on how the cell decides to repair that cut. If the cell uses non-homologous end joining, or NHEJ,
It's a sloppy quick fix. It often introduces small deletions or insertions, which usually knocks out the function of that gene.
but if the researchers provide a template, the cell can use homology-directed repair, or HDR.
And that is the precise knock-in mechanism. It allows scientists to insert or correct a specific sequence, which is the dream for gene therapy. The diagram for this is one of the clearest in the source material, showing that fork in the road from the double-strand break to the subsequent repair pathway.
Okay, next, let's look at the classic blotting procedures. These separate and identify macromolecules based on size, and we rely on the mnemonic SNOW-DRP.
Right, SNOW for Southern, Northern, Western, DRP for DNA, RNA, protein.
So Southern blot is used for DNA, using a DNA probe.
Right. It's useful for identifying specific fragment sizes, which helps diagnose diseases characterized by repeat expansions or restriction fragment length polymorphisms, like the CDG expansion you see in Fragile X.
Northern blot is used for RNA. And because RNA levels reflect mRNA levels, the northern blot gives you information about gene expression.
Exactly. And Western blot is used for protein, using an antibody probe. This is the gold standard for identifying and quantifying specific proteins in a sample. It's often used as a confirmatory test, for example, to confirm the presence of viral antigens, like in an HIV test.
And finally, flow cytometry. This is an essential technique for assessing cell characteristics, particularly in immunology.
The flow cytometry diagram is elegant. It analyzes cells that have been tagged with fluorescent antibodies, say antibodies against CD4 or CD8 T-cell markers, as they pass single file through a laser beam.
And the resulting light scatter forward scatter for size and side scatter for granularity allows researchers to accurately plot and count distinct cell populations.
Exactly. This is how we monitor critical immune populations, like CD4 positive T-cell counts in HIV patients. It gives you a direct, quantifiable measure of the health of the immune system.
Okay, now we enter the core energy economy, metabolism. This section of the source material is just dense with diagrams of cycles and regulatory points. These are essentially the main levers the body uses to adjust metabolic flow.
And we cannot overstate the importance of the rate-determining enzymes. These are the single most important regulatory points in any given pathway.
Oh.
Think of them as the control valves.
Like PFK1 and glycolysis.
Isocitrate dehydrogenase in the TCA cycle, HMG-CoA reductase in cholesterol synthesis. These are the control valves that govern the entire flow of energy through those systems.
So let's start with glycolysis and its regulation, with a focus on the diagram. It's the ancient pathway for breaking down glucose. The net process is simple. Glucose plus 2 ADP and 2 NAD plus yields 2 pyruvate, 2 net ATP, and 2 NADH. But the initial steps are governed by tissue specialization.
Yes, and we must contrast hexokinase and glucokinase. This is a brilliant example of how the body uses subtly different enzymes to achieve fundamentally different goals in different organs.
Okay, so hexokinase is found in most peripheral tissues. It has a high affinity for glucose, a low Kbem, so it acts like a vacuum cleaner. It efficiently grabs any glucose available, even when blood sugar is low.
Right, but it has a low maximal capacity, or Vmax, and it's strongly inhibited by its own product, glucose-6-phosphate. It's all about prioritizing the needs of the local cell first.
Glucokinase, however, is the liver and pancreatic beta cell enzyme, and it's the opposite. It has a low affinity, a high Knem, but a high capacity, a high Vmax.
And it's not inhibited by its product. Its activity is induced by insulin. So the liver uses glucokinase to store glucose efficiently only when blood concentrations are very high, like after a big meal. It's a floodgate, not a vacuum cleaner.
The ultimate control point in the middle of glycolysis is phosphofructokinase loss 1, or PFK1. And the source provides this critical regulatory diagram that revolves around fructose 2-cymbal-6-bisphosphate.
Yes, F26BP is the master regulator. It acts as the body's internal signal of abundance, and its level is controlled by a single bifunctional enzyme, PFK2 FP-PASE2. This is the metabolic light switch.
Okay, detail the switch for us. In the fed state, when you've just eaten.
Insulin is dominant. High insulin activates the PFK2 side of that enzyme complex. PFK2 produces F26BP, and that F26BP then strongly stimulates PFK1.
And the result is glycolysis speeds way up, burn the fuel, store the rest.
Exactly. Conversely, in the fasting state, glucagon or epinephrine dominates. These hormones activate the other side of the enzyme, FP-PASE2, through phosphorylation. This decreases the level of F26BP.
And removing that signal inhibits PFK1.
It slows glycolysis way down, and critically, it allows the pathway to reverse, favoring gluconeogenesis, the process of generating new glucose from non-carbohydrate sources. That one switch determines the feed of glucose in the liver.
Once pyruvate is produced, it has to be converted to acetyl-CoA to enter the TCA cycle. This is handled by the massive pyruvate dehydrogenase complex, or PDC, which links carbohydrate metabolism to the core energy hub.
And the PDC requires 5 essential cofactors. They're all B vitamins or their derivatives: B1, thiamine; B2, riboflavin; B3, niacin; lipoic acid; and B5, pantothenic acid. The complex is activated when the cell signals low energy, usually indicated by a high NAD plus NADH ratio.
A deficiency in the PDC, which is often an X-linked condition, is neurologically devastating and causes severe lactic acidosis.
Right. The pyruvate, which is unable to form acetyl-CoA, gets shunted into lactate and alanine instead. To manage this, the treatment involves giving high doses of B1 thiamine and providing ketogenic nutrients, specifically the amino acids lysine and leucine, which can be broken down directly into acetyl-CoA without needing that defective PDC complex.
Okay, let's move to the central cycles. The TCA cycle and the electron transport chain.
The TCA cycle, or Krebs cycle, is the main metabolic hub, and it happens inside the mitochondrial matrix. Acetyl-CoA condenses with oxaloacetate, or OAA, to form citrate, and that initiates the A-step cycle.
And the cycle isn't just about energy, it's amphibolic. It provides precursors for biosynthesis while it's generating high-energy electrons. But focusing on energy, the yield is substantial. For every acetyl-CoA that enters, we get 3 NADH, 1 FADH2, and 1 GTP.
Which translates to about 10 ATP equivalents per turn. And the regulation centers on the irreversible enzymes: citrate synthase, isocitrate dehydrogenase, and alpha-ketoglutarate dehydrogenase. And notice how citrate, the very first product, acts as a long-range signal.
How so?
It actually inhibits PFK1 way back in glycolysis. It's a feedback loop. If the TCA cycle is full of citrate, that tells the cell it doesn't need to break down any more glucose right now.
So all those high-energy electron carriers, NADH and FADH2, then proceed to the electron transport chain, or ETC, and oxidative phosphorylation. This is in the inner mitochondrial membrane. This is where the bulk of life's energy is harvested.
The ETC diagram shows this spectacular process. Electrons from NADH and FADH2 are passed down through the 4 complexes, I through 5e. And the energy from that transfer is used to actively pump protons, H+ ions, from the mitochondrial matrix into the intermembrane space.
So you're generating this massive electrical and chemical gradient.
It's a huge potential energy gradient,
Right.
and that is the money shot. The protons then flow back into the matrix through ATP synthase complex V, which acts like a tiny turbine. It uses the mechanical force of that proton flow to phosphorylate ADP into ATP.
And the yield is defined. One NADH gives you about 2.5 ATP, while one FADH2 gives you about 1.5 ATP.
And this leads to a fascinating clinical insight: uncoupling agents. Agents like toxic doses of aspirin or the natural uncoupling proteins found in brown fat can disrupt this process.
So what do? They poke holes in the membrane.
That is exactly what they do. They poke holes in the intermitochondrial membrane. The protons flow back across the membrane without passing through ATP synthase. So the potential energy of the gradient is just released as heat.
So you're not making ATP, you're just making heat.
You got it. And that's why a toxic aspirin overdose causes hyperthermia, and it's why brown fat is so essential for thermogenesis in newborns.
Okay, the body also has specialized routes for glucose, like the pentose phosphate pathway, also called the HMP shunt, which occurs entirely in the cytoplasm.
This pathway has two non-negotiable purposes. First, it produces ribose 5-phosphate, which is essential for synthesizing nucleotides for DNA and RNA.
And most crucially, it produces NADPH.
Yes. NADPH is vital. It's required for all reductive biosynthesis reactions, like making fatty acids and cholesterol. And especially in red blood cells, it's needed to maintain glutathione in its reduced state. Reduced glutathione is the cell's frontline defense against oxidative damage from free radicals.
So failure of this pathway causes glucose 6-phosphate dehydrogenase, or a G6PD deficiency. This is the most common human enzyme deficiency worldwide, and it's an X-linked recessive disorder.
Right. Without adequate G6PD, the cell can't produce NADPH fast enough. So when the cell encounters oxidative stress, it can't detoxify the peroxides. This leads to oxidative damage to hemoglobin, which precipitates and forms these characteristic inclusion bodies called Heinz bodies.
And that leads to hemolysis, the destruction of red blood cells.
Exactly. And the hemolysis is triggered by specific oxidative insults-consuming fava beans, which is called favism, taking certain oxidant drugs like sulfonamides, or even just having a systemic infection. The prevalence of this trait in malaria-endemic areas is a perfect example of natural selection, as G6PD deficiency seems to confer some protection against severe malaria.
We also need to detail specific disorders of monosaccharide metabolism. These show how minor enzymatic defects can lead to major systemic problems.
Let's compare the two main fructose disorders. Essential fructosuria is a benign condition. It's caused by a defect in fructokinase. Fructose simply accumulates harmlessly and spills into the urine. No big deal.
But hereditary fructose intolerance, a defect in aldolase B, is severe.
It is. Because aldolase B is blocked, fructose 1-phosphate accumulates. And this toxic intermediate traps intracellular phosphate, locking it up. This lack of free phosphate inhibits critical processes like glycogenolysis and gluconeogenesis.
So clinically, this leads to severe hypoglycemia, jaundice, and eventual cirrhosis if fructose isn't strictly eliminated from the diet.
Correct. And it's a similar story in galactose metabolism. Galactokinase deficiency is relatively mild, mainly just causing infantile cataract.
But classic galactosemia
Is caused by a defect in galactose 1-phosphate uridyl transferase, and is toxic. The accumulation of galactose 1-phosphate causes failure to thrive, hepatomegaly, jaundice, infantile cataracts, and tragically, it increases the risk of E. coli sepsis in newborns. The mandatory treatment is complete, immediate exclusion of all galactose and lactose from the diet.
Okay, when we break down amino acids for energy, we generate ammonia, which is profoundly neurotoxic. The body has to detoxify this ammonia by converting it into urea for excretion, and this occurs via the urea cycle.
The urea cycle diagram is key. It's an elegant, energy-consuming process that spans both the liver cytosol and mitochondria. It incorporates carbon dioxide and ammonia via carbamoyl phosphate with aspartate, and eventually generates urea and fumarate, safely packaging that toxic nitrogen.
The most common disorder of this cycle is ornithine tranocarbamylase, or OTC deficiency. It's an X-linked recessive condition.
Right. And when OTC is blocked, ammonia cannot be eliminated. This leads to acute, life-threatening hyperammonemia, especially in infants. But critically, when OTC is deficient, the intermediate carbamoyl phosphate accumulates in the mitochondria because it can't proceed in the urea cycle.
So it's a backlog.
It's a huge backlog.
There's
And this accumulation forces the carbamoyl phosphate to shunt into the pyrimidine synthesis pathway over in the cytosol.
And that shunting results in the key diagnostic sign, elevated orotic acid in the blood and urine.
Exactly. It's the ultimate upstream backlog indicator, and it's what distinguishes OTC deficiency from other causes of hyperammonemia.
We also see elegant nitrogen management in the alanine cycle.
Yes. During fasting, muscle tissue needs to break down protein for energy, and that generates nitrogen waste. The muscle sends this nitrogen safely to the liver, primarily packaged as alanine or glutamate.
And the liver then does two things. It performs gluconeogenesis, creating glucose from the alanine backbone, and it runs the urea cycle to excrete the nitrogen. Then it sends the newly made glucose back to the muscle to fuel its activity.
It's a perfect closed-loop system for energy and waste management between the muscle and the liver.
And finally, phenylketonuria, or PKU. This is an autosomal recessive disorder where the conversion of phenylalanine to tyrosine is blocked due to a deficiency in the enzyme phenylalanine hydroxylase, or PAH.
Right, so phenylalanine accumulates to toxic levels and gets shunted into producing phenylketones. If this is left untreated, the consequences are devastating and permanent. Severe intellectual disability, microcephaly, seizures, and that characteristic musty body odor due to the ketone accumulation.
And since tyrosine can't be made from phenylalanine in these patients, it becomes an essential amino acid and has to be supplemented in the diet.
Exactly. The entire treatment hinges on strict dietary restriction of phenylalanine, which means avoiding high-protein foods and artificial sweeteners like aspartame. Neonatal screening is mandatory for this in most countries because early dietary intervention prevents the devastating neurological damage completely.
Now for the fats. Lipid metabolism and transport. Fatty acid synthesis and degradation are not only tightly regulated, but they are spatially segregated to make sure they don't happen at the same time.
Right. Fatty acid synthesis occurs in the cytoplasm, and it requires the citrate shuttle to move acetyl-CoA out of the mitochondria to act as a precursor.
So you can remember cytoplasm and citrate.
Exactly. Conversely, fatty acid degradation or beta oxidation occurs inside the mitochondrial matrix, and it requires the carnitine shuttle to transport long-chain fatty acids inward for breakdown.
Okay. And the lipid transport diagram is one of the most complex but conceptually crucial diagrams in the source material. It traces the fate of dietary fat from absorption all the way to utilization.
Right, so dietary fats are first absorbed in the intestines as micelles, and then they're repackaged inside the intestinal cells into these massive lipoprotein particles called chylomicrons.
And chylomicrons enter the lymphatics, then the systemic circulation, carrying dietary triglycerides and cholesterol.
Once they're in the circulation, the chylomicrons encounter lipoprotein lipase, LPL, on the walls of blood vessels near muscle and adipose tissue. LPL is activated by a tag called apolipoprotein CII, or ApoCI.
And it acts like a molecular garbage disposal.
It does. It cleaves the triglycerides and releases free fatty acids for immediate energy use or for storage.
After the LPL action, the remaining chylomicron remnants, now richer in cholesterol, are taken up by the liver via the ApoE receptor.
Correct. The liver then processes these components and releases its endogenous lipids packaged into very low-density lipoprotein, or VLDL, for internal transport. VLDL eventually sheds its triglycerides in the periphery and becomes LDL, the so-called bad cholesterol.
Which brings us to the importance of the apolipoproteins, the ID tags and regulatory keys on these particles. ApoCI is the LPL activator.
ApoAI is found on HDL, the good cholesterol, and it activates an enzyme called L-sheet, which is required for HDL maturation and reverse cholesterol transport.
And the B proteins are essential for identity. ApoB48 is specific to chylomicrons, which come from the gut.
And ApoB100 is specific to VLDL, IDL, and LDL, which all originate in the liver. And it serves as the crucial ligand for the LDL receptor, determining how cholesterol is cleared from the blood.
And failures in this transport system cause the hyperlipidemias. For instance, type I hyperchylomicronemia is caused by a deficiency in either LPL or its activator, ApoCII.
Right. And this causes a massive backlog of chylomicrons and triglycerides in the blood, which leads to acute pancreatitis.
And type 2 hypercholesterolemia, the most common type, is due to defective LDL receptors or defects in that ApoB100 ligand.
This prevents the liver from clearing cholesterol efficiently, leading to extremely high LDL levels, causing xanthomas and early accelerated atherosclerosis.
Finally, we have to synthesize all these cycles by looking at the fasted versus fed state diagram. This shows the body's coordinated metabolic response across all the organs.
In the fed state, insulin is dominant. Insulin promotes storage and utilization. It stimulates glucose uptake, drives glycolysis, glycogen synthesis, and lipid synthesis. The goal is to quickly process and store that incoming flood of nutrients.
In the fasting or starvation state, lasting from 1 to 3 days, glucagon, epinephrine, and cortisol take over. The priority shifts entirely to maintaining blood glucose for the brain.
This is achieved through hepatic glycogenolysis, breaking down stored glycogen and gluconeogenesis, primarily from substrates like lactate, alanine from that alanine cycle, and glycerol from fat breakdown.
And at the same time, the liver begins producing ketone bodies, acetoacetate, and beta-hydroxybutyrate from fatty acid breakdown.
Yes, and these ketone bodies provide an alternative, crucial fuel source for peripheral tissues and eventually for the brain, which spares a limited glucose supply. It's a beautiful, complex balancing act between conserving resources and generating emergency fuel.
Okay, we close our deep dive by synthesizing these metabolic diagrams into the specific clinical findings they predict, starting with storage disorders. Part 4, clinical connections.
We start with the glycogen storage diseases, or GSDs. These are defects in enzymes needed to store or, more importantly, mobilize glycogen. The failure results in an abnormal accumulation of glycogen, either in the liver, leading to hepatomegaly and hypoglycemia, or in muscle, leading to cramps and exercise intolerance.
Type I is von Gurk disease. It's a defect in glucose 6-phosphatase. That's the enzyme needed for the very last step of both glycogenolysis and gluconeogenesis.
Since the final exit door for glucose from the liver is blocked, the clinical picture is severe. Profound fasting hypoglycemia is the absolute hallmark. You also see a massive accumulation of glycogen in the liver and kidneys, and a buildup of upstream metabolites like blood lactate, uric acid, and triglycerides.
And the constant need to prevent that hypoglycemia requires frequent oral glucose or cornstarch feedings.
It does. Now, type 2 is Pompe disease. The defect here is in the lysosomal alpha-1 glucoglucosidase, also known as acid maltase.
So the accumulation is happening inside the lysosome.
Exactly, which leads to cellular dysfunction. The mnemonic is that Pompe pushes on the heart, liver, and muscle. The resulting organomegaly is dramatic, and the key finding is progressive, severe cardiomyopathy, which typically leads to death in infancy.
And type V is McArdle disease. This is a defect in skeletal muscle glycogen phosphorylase.
So this specific enzyme defect prevents muscle tissue from breaking down its own glycogen stores for use during exercise. Clinically, patients experience muscle cramps, pain, and rhabdomyolysis during exercise, but they often experience a second wind as blood glucose and fatty acids become available from other sources.
Crucially, unlike von Gierke, blood glucose levels are typically unaffected.
Right, because the liver enzyme is entirely normal and can release glucose systemically just fine. The problem is purely in the muscle.
Next up, the lysosomal storage diseases, or LSDs. These are caused by defects in lysosomal hydrolytic enzymes, which results in the accumulation of undigested complex molecules, particularly sphingolipids.
And we need to focus on how this specific accumulation dictates the clinical findings. Let's focus on the classic sphingolipidoses, particularly those that present with that diagnostic cherry red spot on the macula of the eye.
Okay, first, Tay-Sachs disease. That's a hexasaminidase, a defect, leading to the toxic accumulation of GM2 ganglioside.
And this causes progressive neurodegeneration, developmental delay, and the cherry red spot. But what distinguishes Tay-Sachs from other LSDs is that the source explicitly notes it does not cause hepatosplenomegaly. The accumulation is purely neuronal.
Okay, contrast that with Niemann-Pick disease. That's caused by a sphingomyelinase defect, leading to the accumulation of sphingomyelin and cholesterol.
Niemann-Pick also causes progressive neurodegeneration and the cherry red spot. But the presence of hepatosplenomegaly is the key differentiator. Pathologically, it's defined by the presence of these foam cells, which are lipid-laden macrophages found throughout the radiculoendothelial system.
And finally, the most common LSD is Gaucher disease, a defect in glucocerebrosidase leading to glucocerebroside accumulation.
Gaucher disease causes massive hepatosplenomegaly and pancytopenia. And the diagnostic finding is the macrophage that looks like crumpled tissue paper due to the accumulated substrate. These diseases just highlight that even a single enzyme failure can derail complex cellular cleanup systems, often with devastating consequences that depend entirely on where that undigested product settles.
And we'll finish with essential vitamins and clinical correlation. We've established that the entire metabolic economy relies on these key cofactors.
Let's review the essential vitamins and their deficiency syndromes, as detailed in the source material.
Starting with vitamin B1, thiamine, it is a mandatory cofactor for 3 crucial enzymes: pyruvate dehydrogenase, alpha-ketoglutarate dehydrogenase in the TCA cycle, and transcetylase in the HMP shunt. A deficiency basically cripples the ability to use carbohydrates for energy.
Right, and deficiency causes beriberi. Wet beriberi, characterized by high-output cardiac failure due to energy failure, and dry beriberi, which causes polyneuritis. And crucially, in chronic alcoholics, it manifests as Wernicke-Korsakoff syndrome, which starts with confusion, ophthalmoplegia, and ataxia, and then progresses to permanent memory loss and confabulation.
Vitamin B3, niacin, is the precursor for NAD plus and NADP plus I. Its severe deficiency causes pellagra, which presents with the famous 3Ds, diarrhea, dermatitis, often a collar-like rash, and dementia.
And we have to remember that B3 deficiency can be caused not just by dietary lack, but also by conditions that disrupt tryptophan metabolism, since tryptophan is a precursor to niacin. This includes things like Hartnup disease or malignant carcinoid syndrome.
And finally, we have to link vitamin B9, folate, and B12 cobalamin, which are chemically intertwined. Both are essential for single carbon transfers and the synthesis of DNA. Therefore, a deficiency in either one causes macrocytic megaloblastic anemia.
Folate deficiency, which is especially important in pregnancy, is directly linked to an increased risk of neural tube defects. And it's depleted very quickly because the body stores minimal amounts.
B12 deficiency, however, is clinically more severe because B12 is crucial for the conversion of homocysteine to methionine. Without B12, folate gets trapped as methyl THF and can't be used for DNA synthesis. This is the methyl trap hypothesis.
More critically, B12 deficiency leads to unique neurological symptoms that you do not see with folate deficiency. Subacute combined degeneration. This involves the demyelination of the dorsal columns, which carry sensory information, and the lateral corticospinal tracts for motor control.
So this leads to paresthesias, gait instability, and severe neurological deficits that are often irreversible if treatment is delayed.
Exactly. And the absorption of B12 requires intrinsic factor from the stomach, so deficiency is common in conditions that lead to malabsorption, like pernicious anemia, surgery on the terminal ileum, or Crohn's disease.
What an incredible, dense journey we've taken through the machinery of life. I mean, we started with the physical coiling of DNA around histones. We saw how that coiling is regulated by these subtle chemical tags, traced the fate of a sugar molecule from the mouth all the way through the TCA cycle, and examined how a tiny glitch, a single defective enzyme like HGPRT or aldolase B can trigger a massive systemic collapse.
The aha moment here, for me at least, is the realization that these are not isolated facts. They are all pieces of a single grand mechanism. The fact that the same carbamoyl phosphate intermediate that the urea cycle needs for nitrogen disposal can shunt into the pyrimidine synthesis pathway, causing orotic acid buildup. That means nitrogen disposal is intrinsically linked to nucleotide creation. Everything is communicating with everything else.
It is the ultimate system of checks and balances. We've seen regulation at every single level. The structural integrity of collagen demanding vitamin C and copper, the entire energy economy being governed by that F26BP switch that perfectly balances glycolysis and gluconeogenesis, and the genetic blueprint being constantly policed by multiple layers of DNA repair mechanisms like NER and MMR.
The source material shows us that the body's machinery, from the replication fork all the way to the TCA cycle, operates under this finely tuned regulation that constantly balances energy needs with structural integrity. When you consider the sheer number of checkpoints like the P53P21 system regulating the cell cycle, the elaborate protein trafficking zip codes we talked about, or that F26BP switch in glucose metabolism, the important question this raises is this. What fundamental overarching principle prevents all of these finely balanced pathways from collapsing simultaneously, and how robust is that principle truly, given how often we see these single enzyme failures cascade into catastrophic disease?